again. So, well, first of all, thank you for listening to the presentation. And so first, imagine living in an environment that has no light, high temperatures, and high pressures.
Yep.
Living in a hydrothermal vent would feel like. And so what a hydrothermal vent is, they are these areas in the ocean that emit a mixture of seawater and reactive elements. And if you click into my poster, you'll see a picture under the introduction. So I separated the hydrothermal system into two parts. First is the vent. The vent is where the emission is happening. It's the opening where the emission happens.
Mm-hmm.
What comes out is the plume. It's called a plume.
Okay.
And hydrothermal systems, they are unique because one location can actually have several different types of physical and chemical conditions. And that's the types of organisms living there, especially the microbial organisms living there.
Mm-hmm.
The reason why these microbes they are of interest is because they are an important source of carbon for deep ocean animals, and possibly reveal what life on early Earth and even other planets are like. And these enzymes, the enzymes from these microbes, they can have industrial uses. And so because these environments are so extreme and they're so deep in the ocean, normally we wouldn't be able to reach these places. And so studying them is actually a challenge. But that challenge can be solved using metagenomics, which is the study of all the genetic materials in a sample. The reason why is when we use metagenomics, we just collect the material, and we don't need to cultivate that material in a lab or anything, which makes it very convenient and also slightly more unbiased. And genomics is also a very fast and inexpensive process. And the data that we get from the analysis, they're stored in databases that are easily accessible. And one of those databases is the Integrated Microbial Genome Database, or And so for my project, I use IMG to compare 7 hydrothermal vent samples to answer the question of what types of bacteria are living in the hydrothermal vents and what types of carbon, nitrogen, and sulfur processes are in these 7 vents. And so the 7 vents, 5 of them are plume samples from the Western Pacific Ocean. And then there are also 2 Greek vent samples, which I call the red mat and the white mat, because these microbes, they're clustered in like these fluffy mats on the floor.
Okay.
Yeah. And so normally before we do any analysis on these samples, we would have to sequence the genetic material and then annotate the genetic material before we can do any sort of analysis and make conclusions. However, the analysis and the annotations already been done years ago, and the information is all stored in IMG. So all I have to do is access it from IMG, which is the homepage I have shown under my materials and methods section.
Okay.
It contains it's a public database, and it contains various different samples and all the tools necessary to study these samples. And so one of the tools I used was the phylogenetic distribution tool. It helps me see what the abundant classes and phyla of bacteria are in my samples. And this second tool is called the abundance profiling tool. The abundance profiling tool tells me how many genes are there. Like if I wanted to find a certain enzyme, the abundance profiling tool will tell me how many genes in my sample are responsible for that enzyme. And because my samples are different sizes, I had to standardize the gene counts that I got. But from my analysis, it seems like Proteobacteria, which is a major bacteria class, Proteobacteria was the most abundant in all my 7 samples. But just knowing that isn't really enough to say anything about my samples, because when you say Proteobacteria is the most abundant, it's like saying mammals are the most abundant, but there are so many different mammals. And so that's why you have to go deeper and look at the specific classes. And then in my results, I show comparative I show a comparative bar graph of the major proteobacteria classes that were found in my samples.
Ooh.
And so the second thing I took a look at was the carbon fixation pathways. So carbon fixation is that process where carbon dioxide is turned into usable carbons like sugars, and then the microorganisms will use that sugar. And there are 3 carbon fixation pathways that I focused on.
Mm-hmm.
The one that is most common, or the one that people will be most aware of, is the Calvin-Benson cycle, because that's the one that's used by plants. However, the Calvin-Benson cycle is relatively more energy consuming. And so that's why there's also the Wood-Yondahl and the reverse TCA cycle, which require less energy, but they have oxygen sensitive enzymes. So they can only operate in minimal to no oxygen conditions.
Mm-hmm.
From the graph here, you can see that the vents, they rely a lot more on Wood-Yondahl and reverse TCA, which tells you that vents are slightly more anaerobic.
Okay, take me through the graph just a minute. Which graph are you showing?
That would be the middle graph, the graph in the middle.
Okay, I see it now. Yep.
Mm-hmm.
And then the abundance of biomarkers.
And then the 2 vent samples are the last 2, the Santorini red and the Colombo white.
Yep.
And you can see they rely a lot more on Wood-Yondahl and reverse TCA, which are the oxygen sensitive pathogens.
Mm-hmm. Mm-hmm.
And so the last graph, the one on the farthest right, shows the abundance of nitrogen and sulfur processing genes.